Let's try a little thought experiment today. I want you to imagine a tumor. And I really want you to stop picturing it as just, a random chaotic lump of rogue cells.
Right. Yeah, that's such an outdated way to look at it.
Exactly. Instead, picture a complex, bustling city. So you have the tumor cells, sure, but you also have the immune system's police force patrolling the area. You've got the structural builder cells paving roads, and the blood vessels acting as supply lines feeding the whole operation.
That's a whole ecosystem in there.
It really is. Now, if you wanted to understand how dangerous this specific city was, would it be enough to just look at like a spreadsheet of the city's demographics, or would you need the actual physical map showing exactly who is living next to whom?
Well, I mean, it really is the difference between having a basic census and having a highly detailed Google map. And historically, in biology, we've only ever had the census.
Which brings us to the core mission of today's deep dive. We are unpacking this fascinating paper from Nature Communications. It's titled, Graph Neural Networks Learn Emergent Tissue Properties from Spatial Molecular Profiles.
It's a bit of a mouthful, but yeah.
It is, yeah. But basically, we want to figure out if artificial intelligence can take the actual 3D spatial map of a tumor and predict disease outcomes, like better than if it just looked at the cells in isolation.
Right. And to really grasp what these researchers pulled off, we need to talk about this massive shift happening in biology right now. Because for years, the absolute gold standard has been something called single-cell RNA sequencing.
Okay, so that's the census we were talking about.
Exactly. It gives us incredibly high-resolution data about the inner workings of individual cells. But there's a catch. To do it, you have to literally break the tissue apart. You dissolve it, isolate the cells, and run them through a machine.
So you get that demographic spreadsheet, but the map is just completely destroyed.
Yeah, totally gone.
It's like tearing a house apart just so you can count the individual bricks. You know exactly what materials you have, but you have no idea what the house actually looked like or where the doors were.
That's a perfect way to put it. But now, though, we have a whole new field called spatial omics. We're talking about technologies with names like IMC, which stands for Imaging Mass Cytometry, and Codex, which is code detection by indexing.
Right, and these are a big deal.
Oh, they're revolutionary.
Yeah.
Because they allow us to measure dozens of proteins in single cells while keeping the tissue completely intact. So we get the high-resolution molecular profile of every cell, and we know exactly where it sits in the tissue architecture.
Okay, let's unpack this for a second. The big question of this paper is whether we can feed this insanely detailed spatial map into a specific type of AI. Something called a graph neural network, or GNN, right?
Right, to see if it can predict clinical outcomes, like how severe is a patient's cancer.
Yeah. But before we get to the actual results, I want to make sure we understand the mechanism here.
Yeah.
Because how does an AI actually read a piece of tissue?
Well, it doesn't look at a photograph the way we do. It reads the tissue by transforming the image into what we call a spatial proximity graph.
Got it. So for you listening, I think the best way to visualize this is a giant connect the dots puzzle.
Yeah, that's exactly what it is.
Imagine every single cell in that tissue image is a dot. In mathematical terms, the researchers call that dot a node. If two cells are sitting right next to each other, like within a specific, strictly defined distance.
Well, they call it fixed kernel radius, yeah.
Right, the fixed kernel radius. If they're within that distance, the AI draws a line between them. And that line is called an edge.
And, those nodes aren't just empty dots either. They are loaded with deep biological information.
Like what kind of information?
Well, depending on the specific experiment, the researchers either loaded the nodes with the full molecular gene expression profile of that specific cell, or they just gave it a simple category label. Like, this is an immune cell versus this is a non-immune cell.
Okay, so the AI builds this massive interconnected web of cellular relationships. And to test if this web helps predict cancer severity, the researchers set up a multi-model ablation study.
Right, which sounds complicated, but it's really just a scientific bake-off.
Exactly. For anyone unfamiliar, you basically take different ingredients or pieces of data away from the AI to see what actually matters. In this case, they pitted 3 different levels of detail against each other.
So competitor number 1 is the bulk model.
The simplest approach.
Yeah. It just looks at the average molecular expression of all the cells in the entire tissue combined into one big smoothie.
Okay, one big smoothie. And then competitor number 2.
That's the single-cell model. This one uses a multi-instance learning algorithm. It
Wow.
looks at the high-resolution data of every individual cell, but it treats them as totally disconnected from one another.
So it has the full census, but no addresses.
Precisely. And finally, you have competitor 3, the spatial tissue architecture. This is the heavy hitter, the graph neural network.
Right, the GNN.
Yeah, it gets the full molecular data and the complete connect the dots map, showing exactly who is interacting with whom.
Now, I have to jump in here and just play devil's advocate for a second, because it seems almost too obvious that the spatial map should win this bake-off.
Oh, for sure. Everyone thought so.
Right. Going back to our city analogy, if you're trying to predict a neighborhood's crime rate, knowing where the troublemakers are hanging out has to be more useful than just knowing they exist somewhere in the city limits, right?
It is highly intuitive. I mean, most people looking at this setup would assume the AI that sees the full interactive architecture of the tissue would absolutely crush the models that just look at broad averages or isolated cells.
Let me just pause here and make sure I'm capturing this setup for everyone listening, just to do a quick recap.
You
So,
know.
bullet 1, we've got this new spatial omics tech that preserves the where of biology. Bullet 2, we are turning tissue images into literal social networks of cells using GNNs. And bullet 3, we are setting up a battle to see if the AI should rely on the overall average of the tissue or the complex spatial map to predict cancer severity. So I want to ask you listening right now, if you were the AI, which one would you rely on? Because let's look at the first real-world clinical case they tested.
So the researchers started with breast cancer. They wanted to see if these models could accurately grade breast cancer tumors. And they used 2 prominent IMC datasets.
Okay, what were the data sets?
One is called IMC Jackson, which has 350 patients, and the other is IMC Metabrick with 454 patients.
Okay, so a few hundred patients each.
Yeah, and the clinical task was for the AI to look at the tissue and predict if the tumor was grade 1, grade 2, or grade 3. Grade 1 being the least aggressive and grade 3 being the most dangerous.
Got it. But how did they measure if the AI was actually doing a good job? Like, what was the grading system for the AI itself?
They use a metric called AUPR, which stands for the area under the precision recall curve.
Okay, I know researchers absolutely love their statistical acronyms, but what does that actually mean in plain English?
Think of it as a way to stop the AI from cheating.
Cheating how?
Well, in medical data, you rarely have an even split. You might have a data set where, say, 80% of the tumors are grade 2. A lazy AI could just guess grade 2 every single time and be 80% accurate without actually learning any biology at all.
Oh, wow. Yeah, that makes sense. It just games the system.
Exactly. So AUPR accounts for those class imbalances. It forces the model to prove it can genuinely identify the rare cases just as well as the common ones.
Okay, that makes total sense. So we have the metric, we have the data sets. Let me guess. The incredibly detailed 3D Google map completely destroyed the basic average smoothie.
Well, here is the massive twist. It completely lost.
Wait, what? Are you serious?
Dead serious. The graph neural networks trained on those intricate spatial graphs did not significantly outperform the simpler models trained on isolated single cells.
That is wild.
And it gets even more shocking. For most of the data sets, they didn't even beat the bulk models, the ones that just looked at the overall average of the tissue.
Okay, hold on. The basic average spreadsheet beat the state-of-the-art social network map. How is that even mathematically possible?
It all comes down to a universal problem in machine learning, which is data set size. We have to remember that spatial omics is cutting edge, meaning these data sets currently only have a few hundred images.
Right, you mentioned 350 and 454 patients.
Exactly. And graph neural networks, on the other hand, are incredibly complex and immensely data hungry. So when you feed a wildly complex AI a tiny amount of data, it does something called overfitting.
Okay, let's unpack overfitting. What does that actually look like for the AI?
It means the model has so much processing power and so many parameters it can tweak that instead of learning the general universal rules of what makes a tumor aggressive, it basically memorizes the specific random noise in the training data.
Oh, I see.
Yeah, it learns the exact quirks of those 300 specific patients. But the moment you show it a new patient, it falls apart because it focused on all the wrong details.
Okay, it gets completely lost in the weeds. It's like trying to learn what a dog is, but you only look at 3 Dalmatians, so you just assume every single dog must have spots.
That is exactly it. Now compare that to the bulk representations. Averages act as a mathematical smoother. By mashing everything together, they erase all that noisy, random, cell-to-cell variability and zero in on the dominant biological signal of the whole tissue.
So it forces it to see the big picture.
Right. And with only a few hundred patients, that strong, smoothed-out signal was robust enough to match or beat the hyper-detailed spatial map that was just, tripping over its own complexity.
So if you're listening right now, you might be thinking, well, why bother with this expensive mapping technology at all? If small sample sizes cause the complex AI to overfit and simple averages smooth out the noise and win the game, maybe mapping cellular neighborhoods is just a colossal waste of time.
Yeah, you would definitely think that at first glance.
But I know there is more to this story, because otherwise this paper wouldn't be in Nature Communications.
Oh, there is a much bigger story here. Because if GNNs didn't win the raw accuracy game, why are researchers still so excited about them? Because when they looked under the hood, they realized the GNNs were learning actual underlying biology, not just playing a statistical math game.
Okay, here's where it gets really interesting. Remember, the AI was trained to do a rigid categorization task, right? It was told to put the tumor in a specific bucket, grade 1, grade 2, or grade 3.
Right.
But the AI doesn't just instantly spit out an answer. It goes through intermediate steps. The GNN uses a process called node pooling to squish all that massive cellular data into a single, unified mathematical representation for the whole tissue.
And when the researchers analyzed how the AI naturally organized these representations, they didn't see rigid buckets. They saw a continuous graded manifold.
Wait, how did they even see that?
To see this, they used something called principal component analysis, or PCA.
Okay, another acronym. Let's break that down.
PCA is essentially a mathematical tool for taking highly complex, multidimensional data and finding the strongest underlying storyline, or axis of variation. Imagine shining a light on a complex 3D object to see its shadow. PCA finds the angle that gives you the most informative shadow.
Oh, I love that analogy. Okay, and what did this shadow look like for these tumors?
When they looked at that first principal component, PC1, which is the main storyline the AI found, it perfectly ordered the tumors along a trajectory.
Really?
Yeah. It placed grade 1 at one end, grade 2 in the middle, and grade 3 at the far end. The AI figured out entirely on its own that cancer isn't just a set of disconnected boxes. It's a gradual evolutionary progression.
That is, wow. And it didn't stop there, did it? Because this continuous trajectory, this PC1, didn't just order the tumor grades. The paper says it correlated strongly with disease-free survival. The concordance index was consistently hitting around 0.6 to 0.7.
Which is a huge deal in this field.
I have to pause you there, though, because to a layperson, getting a 60% or 70% sounds like a D-. Why is a 0.6 concordance index impressive?
Because in survival statistics, a concordance index of 0.5 is a literal coin toss. It's absolute random guessing. Hitting 0.6 or 0.7 means the model has found a genuine predictive biological signal.
So it actually worked.
It did. It learned how deadly the tumor was and how long the patient was likely to survive without a relapse, even though no one ever explicitly programmed it to predict survival. It figured it out organically just from analyzing the spatial map.
That is wild. It basically taught itself oncology.
It kind of did, yeah.
Let's do another quick recap for everyone listening because we're throwing a lot of concepts out there.
Good idea.
So bullet 1, breast cancer grading was tested using 3 levels of data detail. Bullet 2, bulk averages actually tied or beat complex spatial models because small sample sizes caused the AI to overfit. But bullet 3, the GNN naturally mapped the biological progression of cancer anyway, predicting survival rates on its own.
Perfectly summarized.
So, if you were wondering if spatial mapping is a waste of time, hold that thought, because the AI was hiding another secret, wasn't it?
It was. And this brings us to another incredible feature of these specific AI models, which is interpretability. We can actually ask the AI how it's coming to these conclusions.
Right.
The researchers used a specific flavor of GNN called a graph attention network, or GATE.
And the key word there is attention.
Exactly. A GATE doesn't just blindly look at the connections between cells. It assigns attention weights to figure out which interactions are actually driving the disease.
Oh, wait, I think I have a way to picture this. Think of it like being at a really crowded party. A basic algorithm just sees who is standing next to whom in the room. But a graph attention network figures out who is actually having a meaningful conversation and who is just awkwardly standing near the dip.
Yeah, that's exactly it. It highlights the voices that matter. So in the tumor city, the GATE identifies which cell types are actively socializing and driving the cancer forward.
So what did it find when they looked at the breast cancer data?
When they cracked open the GATT model trained on the Metabrick dataset, they looked closely at the attention weights between neighboring cell types. They found that in grade 1 tumors, the AI was focusing its attention heavily on fibroblasts interacting with tumor cells.
Fibroblasts, those are essentially the structural builders of the tissue, right? They lay down the scaffolding. So in early cancer, the tumor is heavily socializing with the builders.
Yes. But by grade 3, the most aggressive and dangerous stage, the AI's attention shifted completely. Now, it was heavily weighting the interactions between tumor cells and macrophages.
Macrophages.
Which are a specialized type of immune cell.
They're the bouncers, the immune guard.
Exactly.
So the AI revealed this hidden dynamic. As cancer gets worse, it stops hanging out with the structural builders and starts engaging directly with the immune guards, likely to suppress them or trick them into protecting the tumor. The AI saw the neighborhood dynamics completely changing.
It did. It showed us exactly how the tumor's social network evolves.
Okay, this completely blows my mind for breast cancer, but I have to wonder, is this neighborhood effect a universal law of how our bodies fight disease, or is this just a quirk of breast tumors?
That's the million-dollar question. Yeah. And to find out, the researchers moved to a completely different battleground, which was colorectal cancer.
Okay, switching gears.
Yeah. They used a Codex dataset this time. And the clinical task was different. They wanted to look at a tumor and predict its anatomical label.
Which means what, exactly?
Specifically, they wanted to know, is this region a tertiary lymphoid structure, known as a TLS, or is it a diffuse immune infiltrate, known as a DII?
Okay, why does that distinction matter for a patient?
It is clinically vital. A TLS means the immune cells have formed a highly structured, organized camp inside the tumor. They are coordinating an attack. That usually points to a much better prognosis for the patient.
Okay, so structured camp is good.
Right. A DII, on the other hand, means the immune cells are just scattered randomly and diffusely throughout the tissue. There's no organized camp, which generally means a much worse outcome.
Okay, so to make this a fair test, the researchers really simplified the map, right? They stripped away all the complex protein data and just labeled every node, every dot in the graph as either immune or non-immune. That's it.
Just a binary label, yeah.
And then they set up a showdown between two different graph algorithms, a GIN, which stands for Graph Isomorphism Network, versus a GCN, a Graph Convolutional Network.
And this is where we uncover a massive biological illusion.
Ooh, I love an illusion. What happened?
The GIN absolutely crushed the GCN at predicting the structures. But when they looked at why, the secret was surprisingly simple. It was all about node degree.
Node degree. In mathematical terms, that just means how many connections a single dot has, right?
Right. And in biological terms, it translates directly to how densely packed the cells are.
Wait, just cellular density? You're telling me raw density alone was enough to predict an organized immune camp versus a scattered infiltrate?
Yes. Density was the loudest signal.
But then why would the GCN completely fail to see that? I mean, density is density. Why would one algorithm see it and the other one completely miss it?
It comes down to how their underlying math works. A GCN uses a mathematical operation called message passing, where it normalizes the connections.
Normalizes? Meaning what?
Well, if a cell has 20 neighbors, the GCN divides the incoming signal by 20. It averages everything out, which essentially wipes away the raw density data. It just sees the proportions. But a GIN uses some aggregation. It literally just adds the neighbors up. 1 + 1 + 1. So it preserves the absolute density of the neighborhood.
Okay, I see. Think of message passing like a politician visiting a town. Instead of listening to 100 angry citizens yelling at once, the politician takes a poll and gets the average sentiment of the crowd. It's cleaner, but you lose the raw intensity of the situation.
That's a great way to think about it.
Whereas some aggregation is feeling the full volume of all 100 people screaming at once, it preserves the raw, intense density of the crowd.
Exactly what's happening. But this raises an immediate red flag.
Yeah, I was just thinking that. If density is the only thing that matters, did the GIN just cheat? Did it just count the cells in a circle and completely ignore the spatial map it was given?
Right. That is exactly what any good peer reviewer would ask. Did it actually learn the architecture, or did it just learn how to count?
So how did they test that?
To find out, the researchers used a technique called gradient saliency analysis.
I love this concept. It's basically putting the AI on the witness stand and interrogating it.
Pretty much.
You point to a specific cell and ask the algorithm, why did this exact cell make you vote for a highly structured TLS? So what did the AI say?
When they ran this analysis, they found something remarkable. They discovered that if a cell had a node degree of 10 to 15, meaning it was in a moderately dense area,
Okay.
and its immediate neighbors were more than 80% immune cells, the AI flagged it as a massive predictor for a TLS, that structured immune camp.
Okay, but honestly, that sounds like it's just counting.
It does. But here is the kicker. Here is the absolute genius of the GIN. They took a cell with those exact same stats, a density of 10 to 15 and 80% immune neighbors.
Right.
But this time, this cell was located in a DII, a diffuse scattered region. When the AI looked at this identical cell, it weighted it much lower, essentially ignored it.
Let me make sure I'm wrapping my head around this. The local stats were identical. The immediate neighbors were exactly the same. How could the AI possibly tell the difference?
Because the GIN was perceiving what researchers call higher-order spatial cues. It was looking way beyond the immediate next-door neighbors.
Oh, wow.
It could sense if that specific cell was embedded in a massive T-cell-enriched neighborhood versus a macrophage-heavy neighborhood without even being explicitly programmed to know the difference between T-cells and macrophages.
So it could literally sense the broader vibe of the cellular neighborhood. It understood the context of the entire city block, not just the house next door.
Exactly.
That is incredible. Let's recap this final segment because it is just mind-blowing. So bullet 1. In colorectal cancer, cellular density is the loudest signal for predicting immune structures. Bullet 2. GIN models outperform GCNs by preserving this density through some aggregation.
Right.
And bullet 3, GNNs can perceive invisible higher-order neighborhood contexts. So to you listening, just think about that for a second. How incredible is it that a mathematical algorithm can sense the complex vibe of a cellular neighborhood in a way that is totally different and potentially more profound than a human pathologist staring at a microscope?
It really changes how we think about diagnostics. And if we pull back and connect all of this to the bigger picture, the findings from this paper are a vital reality check for the field, but also a massive beacon of hope.
A reality check because of the overfitting.
Right. Graph neural networks applied to spatial omics might not immediately beat simple average-based algorithms on small datasets. The risk of overfitting is very real. But we have to remember, that's a data problem, not a model problem.
Right. As the technology scales and we get thousands of tissue images instead of hundreds, these models are going to become unstoppable.
Absolutely unstoppable. But even right now, today, with small data sets, they are entirely unmatched at extracting the why and the how of tissue architecture.
Yeah, that interpretability is everything.
Their true immediate value lies in biomarker discovery. They can find hidden spatial patterns, like the shifting proximity of fibroblasts to macrophages we saw in breast cancer.
Yeah.
And they can decode the physical social networks of the tumor microenvironment. They prove to us mathematically that cancer isn't just about mutated cells, it's about mutated architecture.
That is such a powerful way to frame it. So what does this all mean for you listening right now? We started this deep dive by talking about the tumor as a bustling, complex city, and it turns out AI has finally given us the ability to read the Google map of that city.
It's an exciting time for biology.
It really is.
Yeah.
But I want to leave you with a final puzzle to mull over. Think about this. If diseases like cancer rely so heavily on the spatial organization of their cellular neighborhoods, their social networks, what happens when the medicines of the future stop trying to just poison and kill the bad cells directly?
That's an interesting thought.
What if instead our drugs just try to break up their social connections? Could we cure diseases simply by rearranging the architecture and forcing the troublemakers to move out of the neighborhood?
It's a totally different paradigm.
It's a wild thought. Thank you for joining us on this deep dive, and keep exploring the microscopic cities inside us.